Okay, our second speaker today is Berenice Reed-Gitomer. Berenice grew up in Ireland, where she did her training at Trinity College in Dublin. She then was a Fogarty Fellow at the NIH, and then stayed as a senior staff fellow at the NIH for 6 years. She then went to UT Southwestern, and in 2002, she was recruited to the University of Colorado, where she's currently part of the Polycystic kidney disease group here in the renal division. And today she's going to tell us about autosomal dominant PKD.
Thank you, Sean. Thank you all for giving me an opportunity to talk about what we believe is some very exciting research related to autosomal dominant polycystic kidney disease. So ADPKD, it's primarily thought about as a kidney disease. If you look at it's a genetic disorder related to mutation in either of two genes, the PKD1 or the PKD2 genes, which encode respectively polycystin 1 and 2. So if you look at the top panel on the right, you see a gentleman here about to undergo bilateral nephrectomy. He's got a greatly enlarged abdomen. You can see him below after removal of the kidneys. And in the bottom panel, you can see these massively enlarged cystic kidneys, and this is the key characteristic of the disorder. So the polycystins form a heterodimer complex, and it localizes to an organelle called a primary cilium on the cells. And even though the genes that encode these proteins have been known for more than a decade, unfortunately, we still do not completely understand the function of these proteins. They're thought in the cilium to play a mechanosensor role, but all of the details related to this is quite honestly unknown. However, it's frequently not recognized that PKD is a systemic disorder. Cysts can occur in many organs. They can occur frequently in the liver, the pancreas. Vascular abnormalities are very common, just as Carrie spoke about. You do see vascular dysfunction. Patients tend to have low FMD, and that has been shown even in children who have completely normal kidney function by one of my colleagues, Dr. Kristen Nowak in our group, showed that this occurs in children. The youngest child was 8 years of age. So even though these kids are apparently still normal, there's obviously many defects that are ongoing. So what was the evidence that there might be a mineral bond defect in ADPKD? So first of all, let me explain what I mean by a mineral bond defect. Patients with chronic kidney disease due to multiple different causes later in the stages of disease tend to have bone-related problems. Fracture rates increase, so these are obviously a cohort of patients with low renal function. The polycystin proteins are expressed on the bone-forming cells. These are the osteoblasts and osteocytes. Both of these cells also have a primary cilium. And interestingly, these cells, the osteocytes, produce a growth factor called fibroblast growth factor 23. Studies in animal models have shown that knockout of the PKD1 gene in mice results in a phenotype associated with reduced bone mineral density, trabecular volume, and cortical thickness. In ADPKD patients, the levels of FGF23 are very high. they're much higher compared to patients with chronic kidney disease resultant from other causes. Even when renal function remains still normal, you can detect an elevation in FGF23. So if I direct your attention to the panel on the right-hand side, this is a study where we looked at a large cohort of PKD patients, about 700 PKD patients, and we compared them with over 3,000, nearly 4,000 participants in the chronic kidney disease RIC study. So normally what happens with the progression of chronic kidney disease, you see an ongoing increase in alkaline phosphatase. If you look at the red line on the bottom, this is PKD patients, and you do not see that change in alkaline phosphatase. So there appears to be lower alkaline phosphatase production. So taken together, we hypothesized, and I will tell you that this is a still ongoing study, so I'm going to talk about some of our preliminary results where we don't have the whole picture yet, but so we hypothesized that there may be a bone mineral defect in patients with PKD who characterized by a low turnover state compared to healthy normal controls. And because of the observation that the FGF23 levels are very high, and the fact that this is produced by the osteocytes, we also hypothesize that there may be a dysfunction in the osteocyte. So the aims of our study were twofold, really: to assess bone histomorphometry in adults with ADPKD to determine if they had a primary bone defect compared to healthy volunteers. And secondly, to try and assess osteocyte function. And to do this, we used immunohistochemical staining to look at both FGF23 in the osteocytes, dentin matrix protein 1, which also regulates FGF23, osteopontin, and sclerostin. DMP1 and osteopontin have critical functions during bone mineralization, and sclerostin also regulates osteoblastic function. So our methods are here. We collected serum and urine on the day that we performed a bone biopsy. These were for measurement of renal function, eGFR, calcium phosphate, vitamin D metabolites, alkaline phosphatase, and FGF23. So for the bone biopsy, which is performed on the iliac crest, we used a procedure where patients undergo a double tetracycline labeling protocol, and I'll show you what the purpose of that is in a minute. The bone biopsies that we obtained go through many different analyses. We undertake micro-CT, histomorphometry, scanning electron microscopy, immunohistochemistry, and to look at the mineral composition of bone, FTIR, Fourier transformed infrared imaging. This is a slide where I will show you some of our preliminary data. We do have more data that I'll talk about in a few minutes. This is the first cohort where we looked at all of the urine and serum mineral metabolism related factors. And I'd like to draw your attention to two things. One is that renal function measured by GFR is essentially normal in these patients. As we would expect, we've shown previously, alkaline phosphatase was lower. And lastly, FGF23 levels were slightly higher compared to the controls. So these are patients with very early ADPKD. There is no impairment at this point of their renal function. So in order to do the histomorphometric analyses, This is going to give us a window showing us what exactly is happening in bone, how is bone turnover being regulated. So bone is a very dynamic organ. Normally, it continues to both resorb and resynthesize itself, and this is important to maintain good quality and healthy bone. So if you look at the top panel here, we see the resorption process undertaken by osteoclasts. There's a reversal phase, and then a resynthesis phase by the osteoblasts. So in healthy bone, the resorption and the formation should be matched, so there's no net loss or gain of bone. If there is a slight discrepancy between the rate of resorption and the rate of formation, as shown in the second panel, you're going to have overall net bone loss. And again, in the latter panel is showing a very severe deficit. So, as I explained before, we undertake a double tetracycline labeling procedure where the patients take, or the volunteers take, 2 courses of tetracycline. They're separated by about 14 days. And this is important because if you look at the distance between the 2 labels, measurement of this distance enables us to calculate synthetic parameters that are ongoing in the bone. So tetracycline is deposited in the newly mineralized bone. So we can estimate both static and dynamic indices in the bone. And these are shown in this next table. So this is data from 16 of our patients. They're still fairly young, mean age of 33 years. Renal function is good, GFR about 96. And underneath, I'm showing you in the red box some of the bone parameters that are significantly different. So OV over BV means osteoid volume over bone volume. And you can see compared to normative values from both males and females, it's much lower in the patients with PKD. Osteoid thickness is similarly decreased. Osteoblastic surface, this is the number of the bone-forming cells on the surface of the bone, is decreased. Eroded surface, which is a resorbed bone, is also decreased. The mineralizing surface is highly decreased compared to normal controls. And here one of our dynamic parameters, bone formation rate, is very much lower in the PKD patients. Lastly, mineralization lag time. This is the length of time it takes the osteoid to completely undergo mineralization, is much increased. This is just a more graphical presentation of that data. Anything below the normal reference line is decreased, so most of the bone parameters are decreased. You can see then mineralization lag time is increased. So looking at the histomorphometric indices, which is what I've just shown, these are derived mainly from the trabecular bone. So the trabecular bone is the mesh-like bone that exists between the 2 outer bone cortices. This is a picture of one of our bone biopsies. And one thing that we found that was very surprising is that the thickness of the cortical bone is much thicker compared to normal. So between the 2 cortices, here you see the trabecular bone. So our patients with PKD appear to have abnormalities of the trabecular bone and also changes in the cortical bone, which is much thicker. Our orthopedic surgeon who does these bone biopsies, he really, really struggles with some of our patients to actually get that core out, which is indicative of this very thickened cortical bone. So, of course, the next question was, well, you've seen these abnormalities in the bone. What does it mean in terms, what does it mean for the patient? So we looked at bone mineral density done by DEXA scan, and we were very surprised at what we actually found. So the orange bars represent women, and the blue bars are the men. And the two heavier black lines represent standard deviations below normal. So the first line is -1, and the second is -2 or 2.5 standard deviations below normal. A difference of -1 is considered to be osteopenia, -2.5 is osteoporosis. So we found actually that about 36%, we had 14 measurements of the density at the lumbar region, were below at least 1.1 standard deviation from normal. This is very surprising because these are, for the most part, young, healthy patients. They don't have a significant decrement of the renal function. So we do look at bone mineral density at 3 different locations. We look at the lumbar L1, L4 vertebrae. We look at the femoral neck, and we look at the distal third of the radius. So the difference between these bone sites is in the proportion of trabecular bone to cortical bone. The lumbar vertebrae have a higher proportion of trabecular bone, and there is a slight difference in the actual scaling of these. The lumbar vertebrae goes down to -4, whereas the femoral neck and the radius are only going down to -1.5. So the take-home message is that you do see the biggest changes in the lumbar region, which is consistent with what we're seeing in the histomorphometric measurements, which are also on the trabecular bone. So more trabecular problems in the PKD patients. So the other thing that we looked at was staining. Actually, this is immunohistochemical staining undertaken on the bone biopsies. In the upper panel, you see normal healthy controls. The bottom panel are the PKD patients. I don't know if it projects very well, but so we find higher circulating FGF23 levels, and that's consistent. If you look at the bottom panel, you're looking at the red staining. There appears to be more red staining in the ADPKD patients, consistent with increased formation of FGF23 in the bone. DMP1 regulates FGF23, so we would have expected with high FGF23 potentially lower DMP levels. That isn't what we found. We found that DMP1 dentin matrix protein 1 levels are actually increased. Osteopontin is also increased in the PKD patients. And here is just a graphical presentation of the data. You can see significantly higher FGF23, DMP1, and osteopontin. Another protein, sclerostin, which also regulates bone synthesis. While the mean level was increased, we didn't reach statistical significance with the small number of patients that we have looked at so far. So then to turn to micro-CT, which gives you a 3D picture of the bone architecture. Again, this is looking at the trabecular bone. The large panel is an osteoporotic bone. The first panel here is normal bone. We have started doing bone biopsies in normal healthy controls, so as we can do all of these measurements on them. You can see some slight differences. If you look at this ADPKD bone in particular, you can see that there appears to be an increase in the number of spaces in the bone. So then when you look at the measurements that are made from the micro-CT, surprisingly we found a very slight increase in connectivity. This is the number of connections the struts or the trabecula make within the bone. However, the bone tissue density was significantly decreased in the PKD patients, so this is consistent with what we're seeing by other measures. So, because the osteocyte is the site of formation of FGF23, we wanted to look at the osteocytes in bone, and this was done in collaboration with Dr. Linda Bonewald. Osteocytes reside in lacunae on the bone surface. So the arrows in red indicate normal lacunae, which should be little kind of circles or holes, if you like, in the bone. And the yellow triangles indicate abnormal lacunae. So if you look in the first two panels, you can see what in the cortical bone, the lacunae seem to be almost completely mineralized in. It's apparent also in the trabecular bone, but not as pronounced as it is in cortical bone. So there's definitely abnormalities in the osteocytes in the PKD bone. We don't have a lot of data at this point about the mineral composition of a bone. This is the FTIR data. Here's some preliminary data that was kind of just based on 6 patients, which is what we have before. Very, very small changes in the mineral composition of bone can indicate great changes, if you like, in the structural integrity of the bone. So far, the one thing that we did find a change in was the bone crystallinity. This is the maturity of the crystals, the hydroxyapatite crystals within the bone. And we saw this actually in the cortical bone. The crystal structure seems to be there's higher bone crystallinity in the cortical bone of the PKD patients. So, just to summarize, in ADPKD patients, and again stressing that these patients still have normal renal function, circulating levels of FGF23 are increased while serum alkaline phosphatase is decreased. Bone histomorphometry indicates a low bone turnover state compared to healthy individuals. The bone expression of FGF23, DMP1, and osteopontin are increased, again, compared to healthy controls. Low bone density, especially at the lumbar vertebral region, is detected in 36% of our PKD patients. Micro-CT analysis indicates increased trabecular connectivity but decreased bone density. And we're very excited about the last finding, which is the abnormal osteocyte appearance, at least the lacunae in bone. So FGF23 is a phosphaturic hormone. So it's not kind of unlikely or unusual that you might expect some mineralization changes or mineral content changes in bone. So for the first time, we have described a bone defect in patients with PKD. And it's apparent very early on in the early stages of disease before there's any decrement in renal function. At present, we can't specifically say what the cause. We have many additional experiments that are planned, but it's intriguing to hypothesize that in some way it's related to a primary cilium defect that may somehow trigger osteocyte dysfunction. So our next steps, one of the things that we're trying to do is to collect data, access data from a large cohort of dialysis patients, obviously to determine if there's an increased fracture rate in patients with PKD, because this has never been looked at before. We want, obviously, to go further with our chemical composition experiments looking at difference in FTIR parameters. And hopefully, with collaboration with Linda Bonewall, we will hopefully look a little bit further at the osteocyte structure and function. Many questions, and I don't have answers for them all. Is there an underlying injury in bone that's present in PKD? Is there some kind of communication between the kidney and the bone, or from the bone to the kidney? And whether the Wnt pathway in particular may play a role in these experiments. We have preserved cells for culture from all of our bone biopsies, and these are things that we can undertake in our cell culture. And there's an interesting point of whether hypoxia may have some role by increasing mineralization in bone. We know in the kidney for sure that hypoxia is present. Many of the hypoxic pathways are elevated, and this is thought to be because of as the cysts grow in the kidneys, they impinge the vasculature, reducing kind of oxygen delivery to the tissue. Also, renal blood flow is decreased in the kidney of patients with PKD. So one question, are there changes in renal blood flow in bone of people with PKD? I'd like to acknowledge this is a NIH-funded study that focuses on FGF23. Michelle Chanchel here and Miles Wolfe are my co-PIs on this grant, and we have many collaborators. It takes a village. I think in this case it may take a city to actually answer the questions that we have about bone. So thank you.
That was a great presentation.
Thank you. So in the patients that we have looked at so far, they're PKD1. PKD1, so they have a mutation in the PKD1 gene. It's difficult. There are difficulties. There are probably about nearly 500 or more different mutations that have been identified in the PKD1 gene. So trying to do a specific genotype-phenotype analysis would be somewhat difficult. Many of the mutations are truncating, so they're obviously easier. We're kind of running into that difficulty at the moment with another study where we're looking for modifier genes that may affect severity of disease in PKD, and how you deal with different strengths of different mutations is kind of a tremendous issue. So it's kind of very controversial, and there's one theory that at the germline it's dominant, but it requires a second hit or a second mutation that occurs somatically in the kidney. And the data that's supported that has been mainly that if you look at the number of tubules that actually form cysts, it's a small proportion of the total number of tubules in the kidney. Injury, AKI, can also exacerbate the disease and can exacerbate cystogenesis. This has been shown in animal models as well. However, against that, there are mouse studies that have shown that there's no apparent need for a second hit. We've actually got a grant to try and look at that. to determine whether there's similarities between different cysts within one kidney and cysts between patients. Because if the second hit is random, you're going to get many different mutations, which may also affect disease severity. But animal studies have clearly shown that if you inactivate one copy of the gene, you do definitely get disease. There are hypomorphic animal models that have been developed, and it's very clearly been shown that if you drop the level of the protein below 50%, it does result in cystic phenotype in the kidneys. So dosage is a very important factor in PKD. Yeah. In normal? The normal subjects with normal healthy controls, with normal renal function, there is no increase. In PKD patients with normal function, you do have higher levels. So you would not expect in a healthy control person to ever see an increase in FGF23. Patients with chronic kidney disease, yeah. Correct, absolutely, yes, yes. Uh-huh. Yep.
My question is, at what stage
Oh, okay.
does FGF?
It probably starts to go up in late stage 2, early stage 3, yes. Uh-huh.
And my other question was,
Some of them were, some of them were not. So these are a fairly young cohort of patients. The mean age was 33. Typically, if you can call it typical, age of onset of hypertension is about 34 in men, 32 in women. So we're kind of round about that. But some of them were hypertensive medication. Most typically, they're on an ACE. Sure.